A different way to conduct differential digestion. Differentials are the process involving the separation of male and female cells from evidence collection swabs. This process is very time and labour intensive. Oakland PD aimed to change that. Using an automated process and a degradative enzyme known as DNase 1, this enzyme is used to target any leftover non-sperm DNA that remains after fraction separation. Using DNase 1 for selective degradation, Oakland PD were able to address the following pain points associated with a traditional differential: elongated wash steps, repeated centrifugation steps, and not being able to scale up sample processing capabilities. The process starts with the swabs being placed into a 96 spin-well basket plate and soaked overnight in a PBS solution. They are moved onto the instrument for an initial shaking step before being spinned down. The Versa 1100 then separates the aqueous fractions into tubes and places a droplet into a custom microscope slides for confirmation of sperm. The process is repeated for the epithelial cell fraction. Proteinase K is added to digest the epithelial cells before being spinned down. Again, the Versa 1100 separates the epithelial cell fraction onto slides and tubes to then go on to extraction. Finally, to clean up the sperm fraction, DNase 1 is added to ensure no DNA remaining from the epithelial cell fraction are present. The DNase 1 is then deactivated before the sperm fraction is digested in order to be placed into tubes again to go on to extraction. Differentials, however, can vary from lab to lab. Whilst automating slide preparation may seem like an attractive notion to some, other facilities have moved on to a more streamlined approach known as direct-to-DNA. Essentially, this means diverting the manual process of examining slides and only dealing with a process known as Y screening to determine which samples have male DNA present. Aurora's platform has made it possible to be integrated into whatever differential you currently have employed and validated. Adapters for Promega's Maxwell and QIAGEN's Easy One means the instrument can directly load into your downstream extraction, consolidating multiple workflows onto one single platform. Overall, the DNase 1 workflow bridges the gap of existing sexual assault workflows by allowing you to complete lysis and digestion for a whole 96 samples in a total of around 8 hours start to finish. This is achieved by freeing up an analyst to work on extraction and separation in parallel.